see the screen?
Yep.
Right. I might turn off my video later if my computer gets heated up.
Yeah.
But yeah, otherwise, I'll just let it be. All right, thank you so much for this opportunity to talk to you all. I'm going to talk about some of my work that I did for my PhD, and then I'm going to talk about some work that I did last year as well. But if you look at the image that you have, if you already didn't identify what it was, it's an ant on top of the eye of an Australian bull ant. So this is one of the extreme examples of miniaturization where one species is really tiny and the other species is really large. And so miniaturization is just the evolution of extremely small body size compared to their ancestors. So there's no fixed limit as to when you can call a certain animal to be miniature. It's always relative to some other animal, for example. But it is a very common phenomenon that is seen across animals. So it's seen in reptiles, amphibians, primates, and most importantly, insects. And so I'm going to today talk only about ants and fruit flies. But there are a lot of extreme miniature examples, and fairy flies are one of them. But today I'm only going to talk about ants and fruit flies. So when we talk about miniaturization, it's not exactly the same as what Ant-Man would be. It's just as simple as putting on a suit and becoming a tinier version of yourself, or Alice drinking the drink me potion in the book Alice in the Wonderland, to become an exact miniature replica of herself. Because most often when you want to become miniature, you need to be able to reorganize some of your body plan. And so because of miniaturization, you would see different rates of changes in your organ systems. For example, if you look at some organs, how some of the organs change in size with reducing body size, you can see that systems like circulatory system or musculature in insects scales isometrically, which is basically, it scales with a slope close to 1. But there are a lot of other systems where the slope isn't exactly close to 1. One of the examples is nervous system. And so with decreasing body size, the rate of decrease in the central brain isn't often that steep. So as a result of that, a lot of animals actually have bigger brain relative to their body size. So they seem to retain the brain size or retain the size of the brain as their ancestors when they become miniature. And the other limiting factor that limits how small you can be, how small you can get when you become miniaturized is the size of the reproductive system. So for insects, for example, there is a limit to how small their eggs can be because they need to invest resources in the egg so that it can develop and become an adult. But if there are some insects that actually overcome this limit as well and become, so one of the examples is this mimerid fairy wasps, and so they can get as small as the size of single cellular organisms. And so the way they do it is because they don't really invest as much in the egg size because they're parasitic. So that's how they're able to overcome these limitations. And in addition to that, there's a lot of limitations to...
did you say parasitic or did you mean parthenogenetic?
I would say parasitoid.
Because parasitoids still lay eggs, no?
Yeah, but they don't have to invest a lot in the nutrients that they put in.
Okay, so you just push out the eggs and the eggs will then
develop in the host. Yeah.
Right. Okay.
Right. So, and in addition to that, there's a lot of other limitations to how small one can get. And so, as explained before, nervous system is one of the highly demanding organ system when it comes to metabolic metabolic rate, so it consumes most amount of energy. And so if you want to be able to retain that bigger brain, you need to be able to spend a lot of metabolic, so you will have higher metabolic costs basically. And the other limitation is, for example, is in the size of the neuron. So there's a physical limit to how small the axons of a neuron can be. So if it gets below that particular size, there would be a lot of stochastic ion channel activity. And so that would actually swamp the signal because there would be a lot of noise. And in addition to that, there's also another limit, which is the nuclei. So nucleus inside the neurons and other cells is one of the space-consuming organelles. And so, so you can't really decrease the size of it. So basically, there is a lower limit to how small you can make your cells. But there's again another extreme example of miniaturization where the species of wasp has evolved anucleate neurons. So as it develops from pupae to adult, it actually gets rid of all the nuclei in the neuron, most of the nuclei in the neurons. So apparently maybe they prepare some of the proteins that are required earlier on during the pupal stage so that they can use them in the adult stage. But the reason they can do this is they're very short-lived as well. And we really don't know much about the species as well. And the other limitation for this is the size of the sensory structures. In this case, I'm going to talk about compound eyes. And so there's a limit to how small you can make your eyes. So if your lens is too small, as small as or comparable to the wavelength of the light, your optics would suffer from diffraction artifacts. And the other problem is that if you have really small lens, you'd have a lot of photon noise because you can't collect enough number of photons. And so how exactly with all these limitations, how exactly do animals overcome some of these limitations as they evolve to be smaller and smaller? So one way is that they actually just retain the size of the brain as their ancestors, for example. Even though their body size is smaller, they might have relatively bigger brain. And so as a result, they might have to actually push the brain into different parts of their body. So in this case, there are examples of a beetle doing that, and there are examples of arachnids, some spiders doing that as well. So what happens basically here is that because they have relatively bigger brain relative to their body size, they have bigger brain, they would have to pay higher metabolic costs. But so it gives a meaning, it gives a new meaning to the term thinking on their feet. But one advantage could be that these animals with relatively bigger brain are able to retain some of the behavioral abilities. That's one of the hypotheses. But this kind of adaptation is not exactly possible for a lot of insects, especially when there's a constriction between neck and thorax. So they can't really push their brain from the head towards the thorax, for example, in ants or flies or bees. So in this case, so it has been hypothesized that they just have a smaller brain, but they may actually be, they may have lesser behavioral capabilities than their ancestors, which have bigger brain. So that's the other hypothesis.
Ravi, I had another question,
yeah.
which is, relating to how small neurons can get.
Yeah.
And a sort of key related feature that one must ask about small neurons is their ability to spike at all. Okay. Mm-hmm. Now, in certain creatures, for instance, in C. elegans, which are these nematode worms, they don't have, at least not that we know of, they don't have spiking neurons. I mean, there's a controversy about this. But for the most part, all we see are plateau potentials. Yeah.
Yeah.
And we know that in many insects, there need not be spikes. We can also get neurons which are just sort of of great potential. So
they have neural tricks to overcome the limitations. So they may have smaller neurons, but as I'm aware, it hasn't been really tested.
Yeah, it's really difficult to record.
Exactly. Yeah. And the other thing is also about using group of neurons for multiple behaviors. For example, multitasking is another thing that has been proposed as well. So they may have fewer neurons, but they may be more efficient in the way they interact with each other. But all of these are just hypotheses at this point, as far as I'm aware.
Yeah.
Ravi, Agnesh here.
Yeah.
I have a question. So the circulatory system and the musculature are sort of linearly reducing, whereas the brain is not. That's why we have the relatively bigger size of brain.
Wait, why did you say they are linearly reducing?
The plot that he showed in the
Isometric, basically.
isometrically.
Yeah, they're both linear, but one is isometric. The slope is close to 1.
So I was thinking that there would be a problem, like now the energy supply system is now sort of reduced, but the brain is relatively larger. So how would this tiny insect, and the brain consumes most of the energy. Are there any tricks that these guys use that the brain would require less energy? Is there any study where the enucleate neurons or maybe these graded potentials require less energy, if those kind of mechanisms require less energy?
I don't know if there is any that looks at any study that looks at whether they require less energy, but to answer the relative investment bit, so like I said, there needs to be some body reorganization in the organelle. So they might have relative investment of brain to other systems might be adjusted as well. So what I gave there was for a few species. So it's a slope given for a few insect species. So within a species, you may actually change compared to your ancestor. You may actually change the relative investment of brain to other organelle systems. So one of the examples, maybe not exactly tested in insects, but in humans, one of the hypotheses is that there is competition between digestive system and your nervous system when it comes to metabolic rate requirements. So one of them requires more. So they're both competing. So so you can have differential investment in either of them. But I'm not exactly sure how that happens in insects as such.
I see. Okay. Thank you.
Does that answer the question?
Yeah, sort of. Yeah.
Okay. Okay. So one of the hypotheses, like I just mentioned, is that they may be.
Sorry, Ravi, a quick question before you go ahead.
Yeah.
So isn't it weird that as the size decrease, you see an isometric decrease? Shouldn't this whole volume to surface ratio problem come in, which is that as the body sizes scale up, the number of cells go up cubically, right? So your metabolism should scale up also in a non-linear fashion, right?
Sorry, I don't think I quite got your question.
So what I'm saying is, as your volume scales up, you are going by a factor. You're going as a cube, right?
Mm-hmm.
Your volume scales up faster than. So as the size increases, you would expect that the number of cells would go up as a factor of, not linearly, but as a factor of as a power of 3. So just by that, your metabolism should be scaling up really fast, right? Or at least, I mean, what I'm trying to point out is it shouldn't be linear, right? You should expect some weird non-linearity sitting there, right?
So, if you look at the mouse to elephant curve, the famous mouse to elephant curve, it has an exponent of 0.75, which is a deviation from the expected 2 divided by 3, so 0.66.
Yeah.
Which is the surface area to volume ratio expectation.
Yeah.
Yeah.
Yeah, I get what you're trying to say. So there is a allometric constant, and so a lot of these trends, especially metabolic rate, people have studied, looked at what is this constant metabolic. I forget the exact term, but I'm calling it metabolic constant, or the scaling constant. So it's something close to 2/3. So what you see here is a log scale. So when you take the log, that just goes as a constant. Does that make sense?
But you're saying that the slope is 1.
It's for the log scale.
Yeah,
so,
no. Log scale still, you wouldn't get a slope of 1.
Mm-hmm.
You would get a slope of 2/3, or
that is for the metabolic rate. Yeah.
For the metabolic.
Yeah.
Yeah. What you're showing here is isometric.
So what I'm showing here is for the circulatory system and muscular system volume.
For the brain, yeah.
What I haven't actually told anything about here is the metabolic rate, and I'm sure there are differences in metabolic rate slopes as well when it comes to different organ systems. But again, I don't know if such thing is known in insects. So basically, whether the slope of this scaling for different organ systems would be different or not.
Right.
Yeah.
Tough experiment to do. Yeah.
Yeah. So that I do not know, but as a whole, for an animal, yes. So there's this constant 2/3. So you as you increase the body size, it changes accordingly. Does that?
Okay. Yeah, that makes sense.
Good.
All right. So how does miniaturization really affect behavior? So one of the earliest attempts made to answer this question was done by Cole. And so what he did was he measured the head width of different ants, and then he counted how many types of behaviors do these ants show, and then found a positive correlation. So basically, he just counted so these ants forage. So that's one behavior. These ants fight, that's one behavior. These ants cut leaves, so that's another behavior, and so on. So but the main problem with this approach is that so the way you define and differentiate different behaviors is actually quite subjective. So what you call fighting may actually have a lot of sub-behaviors. So it's really dependent on subjective definitions. And also not all behaviors may be neurally similarly demanding when it comes to neural demands. That's another thing that this particular approach doesn't cover. So we need an objective way to sort of quantify and answer this question about how miniaturization affects behavior.
This seems like a really dubious measure, though. I mean, how do you quantify behavioral repertoire?
Exactly. Yeah. So basically, he just counts the types of behaviors that they. So it's like an ethogram. So you have ants interacting. So they antenate. So he probably writes that as a communication, or if they're fighting, he writes them as fighting or grooming, and so on. But yeah, so definitely this kind of approach really doesn't answer the question that we are posing.
I would have had very serious issues with this if I were a reviewer.
Yeah, it was in 1985, I think.
Yeah, I guess I was too young.
Yeah. But so we need a very good behavior that can be actually quantified objectively, and it's also good if it's comparable across taxa. So we propose visual navigation as one such behavior, because it's a behavior that's known from a lot of insects, and it's very important for central place foragers like ants and bees that have to go back, and wasps as well, that have to go back to a place all the time. And a lot of them are known to use cues such as landmarks or visual panoramas, or maybe even celestial compass cues. So
good to see a bogong moth in here.
Sorry?
Good to see a bogong moth.
Yeah. They're studying a lot about their navigation as well.
Yeah.
Yeah. Right. So in addition to the central place foragers, it's also used by a lot of other insects, fruit flies, for example. So when they have to move to really long distances, they pick a direction and then go straight in that direction for a really long time. And in that process, so they actually use a lot of celestial compass cues. It could be sun, it could be polarized light pattern. And similarly, you've got monarch butterflies that migrate really long distances every alternate generations. And they are also known to use a lot of visual cues for navigation. And then you've got beetles. So when they are making the ball near the dung, there's a lot of competition if you've noticed them. Actually, there's quite a few in Karnataka, quite a few species. So because of that competition, they really need to get away from that dung pile as soon as possible. So the quickest way is to actually travel in a straight line direction away from the dung. And so they use, again, celestial compass cues for navigation. So if they're nocturnal, they've also been shown to use the pattern of the Milky Way galaxy and also moon's polarized light to be able to travel in straight line direction. And in addition to all of this, this is a behavior that is really important for a lot of for success in foraging and mating for a lot of insects. And so, so we think this kind of quantifying and understanding how miniaturization affects visual navigation would be able to answer that question or the test the hypothesis that has been proposed. So before we actually understand how it affects visual navigation, we need to understand how miniaturization affects vision. And ants actually are perfect, and there are a few other insects that are perfect as well, but ants have like a huge range of body size variation across species, and sometimes within the species as well, got a huge range of body sizes that you see within a nest. And in addition to that, corresponding to their head widths, which is a proxy for body size, their eye sizes also vary quite a bit. And so it makes them a really good system to study the effects of miniaturization on their vision. And I'd like to show another plot that I've been working on for a review. So where I compiled all the number of facets or number of ommatidia in the eyes of different arthropods. And so as you can see, Hymenoptera and Lepidoptera have like really huge variation in the number of ommatidia. And similarly, some Coleoptera and Diptera also have some variation. So these orders actually would be perfect to actually test how miniaturization could affect vision.
Ravi, did you do these facet counts yourself, or did you get this from the literature?
I compiled them from the literature, and some of them I've done myself.
I see. There was a software that one of your lab mates had generated to count the number of facets.
Yeah, we use that
right
now. Yeah.
Nice.
But before so I had done this before during my PhD.
Right.
I had done everything manually, which I'll explain. Soon. So we wanted to answer how miniaturization affects vision, but more specifically, we tested two different visual abilities that are important for visual systems. That is sensitivity and resolution or acuity. So you can make a smaller eye in various ways, in different ways. So you can make, you can have fewer but larger ommatidia. That's one way to make a smaller eye. So if you have larger ommatidia, you can actually collect more number of photons. So you have higher sensitivity to light. So you can see in dimmer light, or you can see low contrast images as well. And the other option is to have many but smaller ommatidia. And so that way your interommatidial angle, so the angle that you see here would be really small, and that helps you have higher acuity or resolution, so you can see finer details in the image. And typically there's a trade-off between these two, sensitivity and resolution. So if one increases, the other one tends to decrease. But if you have bigger eyes, so you can actually have some areas where you have better resolution. So you don't have to, and some areas with better sensitivity, for example. But if you have really small eye, what exactly happens? How do they cope with that? Right.
Unless, of course, there is also, something happening at the pigment biochemistry level, in which case you can have both higher sensitivity and higher resolution, right? I mean,
that is correct. And that's something that we find as well, which I'll come to. And in addition to that, all the neural tricks that one can play with, like getting better sensitivity, for example.
Yeah.
But yeah, so that is another option as well. So if you've gotten your eyes tested, you know this chart, but how exactly does one test the visual properties of an eye? And so one of the common ways by which people have quantified these variables is to look at their histology. And so there are certain assumptions that they make about these measures. And so, but you still get an estimate of what their sensitivity and resolution is. But most often, vision is not just about optics, like we just mentioned. It's also about downstream neural properties, and it's got a lot of other mechanisms which can actually help you improve your signal to noise ratio, for example. So we, for that reason, instead of doing the histology, we used a technique called pattern electrorethinography. So this kind of technique has been used in other animals such as rats, crabs, and sharks. So what it does is, so here on the left, you can see a representation of a compound eye and then the optic lobe. So when there is motion across the eye, so the information is relayed from the photoreceptors to the first optic lobe neuropil, which is the lamina. And so if the eye, if the system is able to detect the motion, there would be a change in the voltage in the first optic lobe neuropil, which is lamina, which we can amplify and record. And so that's basically what pattern electrorethinography does. And so the stimulus that we use for this particular technique is, as shown in the corner, which is a contrast reversing sinusoidal grating. So, so basically to quantify the spatial acuity or spatial resolution, we can change the cycles per degree of the gratings and measure at what spatial frequency of the stimulus they stop detecting the motion that you see there. And similarly, to identify contrast sensitivity for each of the spatial frequencies that we tested, we can change the contrast between the black and white bar. And then again, you can look at what contrast value do they stop detecting the motion.
Sorry, I missed this. What is the readout?
I'm going to come to that. Yeah.
Yeah. Okay.
All right. So if I were to give the same test to you, you would probably be able to detect a spacing of these gratings as low as less than 1 mm when you're standing 1 meter from the screen. But as you can see, ants are way below the human legal blindness. But we also know ants vary a lot in body size and eye size. So, so we tested these 4 different species. So we tested these two, the spatial resolution and sensitivity in these 4 species using pattern electrorethinography. But before that, we actually took eye replicas of the ants. The way we do it is we take transparent nail polish and then put a nice layer, very thin layer on the eye. And then when it is dry, we peel it off and then we make cuts at appropriate places and use a cover slip to flatten it. And then we can image it and then manually click on every corner of the facet, which took me forever. But that's how we make this. But again, Sanjay mentioned, one of my lab mates currently has written a software which can do this automatically. But what you see here is that with decreasing head width of the ants, number of ommatidia or facets also decreases. And the size of the ommatidia also decreases, as you can see here. But more importantly, if you notice the insets, so here, what you can notice is that the frontal region of the eye actually has bigger ommatidia compared to the other regions. But as you decrease in size, you tend to lose that kind of specialization. So it's not really clear in the really tiny eye. All right, so the technique as such is, it looks like this, where you have your stimulus that I mentioned about in the beginning, and then we expose this frontal region of the eye of the ants to the stimulus, and we record the voltage response as the ant is looking at the moving stimulus. And so from that, from the voltage value readings, we can estimate the acuity as follows. So we have different spatial frequencies for which we measured the voltage signals, and then we also measured the voltage signal when there's no visual cue, so which is that's my control noise threshold. So acuity is basically the value of the spatial frequency at which the voltage signal goes just above the noise threshold.
Where are you recording from again?
So this is extracellular recording. So we're just touching the eye on
it's some conducting gel and then.
Gel. Right.
So, do you worry that the conducting gel distorts the optics of everything that you're recording?
Right. So that's why we are exposing the frontal region and measuring from the backside.
I see.
Yeah. So the gel is actually not obstructing the frontal region where they're looking at.
Okay.
Yeah. Right. And then again, so to estimate contrast sensitivity, here I've shown two different plots for two different spatial resolutions for one particular ant. And again, you have a control noise threshold, and then the value of contrast at which the signal goes above the control noise threshold is the contrast threshold. That's the value of the threshold. In this case, it's close to 60% for this particular spatial frequency. And the contrast sensitivity is just the inverse of the contrast threshold. So we repeated that for all the 4 species, and then we built a model to see what explains the contrast sensitivity values. So what we find is that with reducing body size, which is correlated with facet size and facet count as well, explains most of the variation that we see in contrast sensitivity in these ants. So basically, size does seem to affect contrast sensitivity in these ants. And the other thing is the acuity. So we again correlated the average facet diameter, which is correlated with body size, with the acuity values for all the species that we studied. And we do see a positive trend, although the rate of increase is not really as much as you see in the contrast values. But still, the smaller species tend to have slightly lower acuity. So another interesting thing about it is that if we compare the values that we get based on the assumptions and based on the optics using various assumptions, those values are actually lower than what we find using this technique, which kind of brings home the point that sometimes optics is not the only, so vision is just not about optics all the time, but it's also dependent on underlying downstream neural properties as well. Right, so to answer the question that we posed.
One second. Yeah. So I'm still a bit not clear about what is the readout, what is this voltage here, where are you recording from? Are you recording from the photoreceptors itself, or are you recording from the first layer of neurons, which is?
So I'm recording from the first layer of the neuron, so it's an extracellular readout. So we plot the frequency distribution of all the frequencies that we get from the readout. And so the first one would be a response from the photoreceptor. And the second one would be a second harmonic of the frequencies. The curve, the histogram would be from the lamina. And so in order to detect movement from, so you have the object here, and if it's here, you should, in order to detect that particular motion, So it is actually detected at the second order neuropil level. So that's the second harmonic voltage value that we're getting. Does that make sense?
I see. So firstly, you're going from the behind, you're poking in into the lamina. Now you're saying that you're removing the first order correlations out with
No,
the assumption that, sorry, go ahead.
Sorry, we are actually not poking. So it's an extracellular readout. So we're just touching the eye using conducting gel, right?
But aren't you touching the behind of the eye?
Yes, exactly.
And you're saying, but you want to, but the input is to the front part of the eye, right?
Right.
So if you're talking about the first layer, isn't that supposed to be, isn't the inputs to the first layer very local in the sense 6 to 10 ommatidia from wherever those ommatidias are looking at, right? So you wouldn't expect signal on the back side for whatever stimulus is being given in the front side.
No, I think it's a pooled response that we are looking at. We're not really looking at one particular group of ommatidia per se.
Would you be able to show a raw trace?
I
okay.
don't have that right now with me.
Okay.
I can share later.
Yeah.
Yeah.
Ravi, I had a question, which is that, with regards to visual acuity, there is several methods out there to determine visual acuity. One is the standard one that Land had established, in which what you're doing is you're making sure the insect is alive, and then you put it on a goniometer and you sort of move it and until you keep track of the pseudopupil.
Right.
And that's one way to do it.
Right.
Then recently in the Lund group, Gavin Taylor came up with this method in which he's looking at micro CT.
Right.
And using, so it's a purely morphological measure of visual acuity, right? Now, as I understand it, Land's method is a good combination of physiology and morphology, right?
Yeah.
In which you're taking account of not just the facet size, etc. You're also looking at the pseudopupil, which gives you some sense of the degree of dark adaptation, the movement of pigments within the ommatidia, and so on. Right. Also would work for instance, the superposition eye. Right. Now, so you have those, and now you're describing a third one, right, in which you're looking at their response to contrast or to moving bars as a measure of visual acuity. Have you compared and contrasted these? And do you find, are you likely to find situations in which the answer from one method is directly opposite to the answer from another method or something like that?
I haven't really seen a directly opposite thing. So the way we did is using Land. So Land has another formula for calculating spatial resolution. And there's also a formula for calculating sensitivity as well. And so they are just, they depend on the morphological characters of the eye. For example, looking at the
just the facet density, right?
Right. So so that's what we've done. So we've calculated those and compared to the values that we find here. And the difference is not really orders of magnitude different, but it is still it still looks significantly different when you look at the values.
I see. So, no, because I mean you're then not including the physiology as part of your measure, which is so it is purely morphologically based measurement. You could do this with a dead insect, for instance.
Yeah.
And that's one way to do it. Except, and for ants, if you're just comparing within ants, then I don't think it matters because they're all apposition. But if you were to do this between Lepidoptera, then actually this would be a huge problem.
Yeah.
Because there, you're now comparing between apposition and superposition, and there is a fair bit of sort of dark adaptation, pigment movement, etc., that changes things.
Yeah. So I'm actually doing that right now. I'm doing a meta-analysis of different values of sensitivity and resolution that have been published in various insects and arachnids. And trying to also compile other variables, as you mentioned, type of the eye and the method that they've used for calculating everything to see which ones, what actually explains. So which ones does the method of measuring actually change the way, the value that you get, or is it the type of the eye, or is it the environmental factors or the other environmental factors that explains the variation in resolution and contrast sensitivity and sensitivity? But you're right. So different methods actually take different things into account. And so that's something that I'm trying to think more in terms of as well, to try to see how different are the values when you use a different method to measure the same thing. And so what is really close to accurate? Yeah, I expect there may not be like orders of magnitude differences in the values, but there might be some differences in the measures using different methods.
Okay.
But yeah. Is that all? Shall I move on?
Yeah, yeah.
All right.
Sorry, interrupting. Yeah.
No, it's all right. So to answer the question, in ants, we do find that with reduced body size, your spatial resolution and contrast sensitivity tend to reduce as well. But now that we know this, how does it actually affect behavioral abilities in ants? And one of the very common behaviors that the ants encounter every time they forage is to be able to detect and avoid obstacles in their path, because even a small twig is actually an obstacle for them, depending on the size of the animal. And so here on the left, you can see a panorama from the human eye point of view, and then a simulation of what an ant with different ommatidia sees the same panorama as. And because the smaller ants tend to have lower resolution and lower contrast sensitivity, we would expect that their ability to detect these obstacles in their path would also be affected. And here on the right, you can actually see, I don't know if you can see it clearly, there's an obstacle there. And as you move towards the obstacle, the size of it also changes, and it's also obstructing a lot more information behind it. And so if the ants are able to detect an obstacle, if they have better resolution and contrast sensitivity, they might actually detour around it earlier in time and before they go really close to the obstacle. So to test that, we studied 5 different species that vary in number of ommatidia, and we trained them to a sugar solution kept on the right side there. And once they have fed and are returning, we kept an obstacle in their path, as you can see. So the ants come all the way towards the obstacle. Sometimes they detour around really quickly, and eventually they go around and go back into their nest. So we filmed this for all 5 different species without an obstacle and with the obstacle. So the without obstacle is on the left side and with is on the right side. And we also defined a corridor, the gray color corridor there. It's just the area between the obstacle, area the obstacle kind of covers from the obstacle to the feeder. And as you can see, so these are just digitized parts from videos. So basically I clicked million times, maybe, I don't know, on every frame, and then created these paths. And you can, as you can see, they almost always go straight towards the nest if there's no obstacle, almost within that particular corridor that I've drawn there. But if with the obstacle, larger animals actually tend to deviate from that corridor earlier on in time and farther from the obstacle compared to really small insects, which go all the way close to the obstacle and then detour around. And so larger ants do detour much farther from the obstacle. And
have you done a non-dimensionalization of this?
Yes, I have a plot, but I didn't actually put that. I thought it would.
And does it tell you anything? Is that?
Yeah, so I plotted the distance from the obstacle at which they start detouring for all these ants, and so we do find a clear pattern of larger ants actually detouring quite earlier, like when they're.
No. What I meant was non-dimensionalizing with respect to, say, the stride length. In other words, is it the case that, ants will deviate if they are within, say, 50 stride lengths from crashing on the object, in each case, in which case you would see something very similar because the stride length of the smaller ant is smaller, it would come closer, but, in fact, they are basically doing the same thing, which is estimating if it's going to take them 50 strides to crash into the object, something like that.
I doubt that they may be doing like a stride count per se, but because
I mean, that's an extreme kind of cartoon example, but the absolute lengths don't really matter in these kind of measurements typically. It's the relative lengths, so if there is
it is the decision making the same.
Correct. So, it isn't, I guess, the object is relatively small or large compared to the animal, right? So if you imagine that this was a very large ant and the object was tiny compared to it, it would just walk over it, not even.
Right.
But
the object is really
very large.
Yeah, but it isn't that large per se. But I understand what you're trying to say. So because the ants are smaller, for smaller ants, the object size might be different at different distances. Is that right?
Yeah. I continue. I just thought it's something worth trying out. Usually, with when you're measuring across size scales, you want to not just look in absolute terms, but also in relative terms. And the way to do that is to non-dimensionalize.
Right.
So that you can get the scale-free effects clearly. Yeah.
We have actually done that for other studies, but I did try giving different sized obstacles in this experiment as well. But I don't remember why I stopped using it after some point. We just ended up using the same obstacle for all of them.
Sure.
But yeah, so in addition to this particular measure, we also have other measures so I've included one of them from the paper, is to quantify where they're looking at as they're walking towards the obstacle or towards the nest. And so we quantified this angle between the nest direction and the direction in which they're looking at. And we plotted histograms of these angles at different distance intervals from the obstacles. And so here I've only shown two species, one of the largest ones and the smallest ones. And 0 degrees here means looking towards the nest. And so as you can see, so for the larger ant, as it goes closer to the obstacle, around 10 to 15, it starts to look slightly away from the nest direction, while the really small one doesn't seem to do as much, and it seems to do when it gets really close to the obstacle, around 5 to 10 centimeters. So that's the other variable that shows that smaller ants actually look away when they're really close to the obstacle as well. Right, so to answer the question.
So, Ravi, there are two factors involved here, right? One is definitely the changes in eye size, right? But the other one is the neural control, the behavior by itself, right? So the assumption here with these experiments is that the behavior is exactly the same, and whatever output or the intention of the behavior or the neural circuits which underlie obstacle detection are exactly the same between ants. And what differs is basically the inputs, right? Your eye size, that's the assumption here. Is there a good reason to assume that? Do we know for a fact that they behave exactly identically?
But not exactly for a particular behavior as this, but I would say detecting obstacles is a very common behavior for all the ants. So it's the same as detecting any object per se. I wouldn't imagine that they would evolve a different mechanism for the same kind of behavior, which is as common as detecting any object, really. I would guess.
No, that's not what I mean. The reaction to the object, that might be different, right? So there's detection, and how it reacts to it. Right? We are assuming that the reaction is exactly the same, which is the implicit assumption here, right?
Right. Yeah, I don't know. I don't know if we know that they react actually differently to an obstacle. Yeah, but actually you can see that in some cases they have. So this one, most of them actually tend to detour around that side. So one of the things that might be happening there is we think might be because they might have another landmark in the background, which they may be using for navigating. So if they have to detour around it, and if they're seeing that particular landmark, for example, like a tree more prominently, they might just use that cue to navigate around. But yeah, that's a good question, but I don't have an answer for that.
Ravi, another question, which is that I presume that these are trained ants, right? Or are they out on their naive round? This is a trained ant.
These are trained ants, but well, I didn't quite mark them all to actually measure how many times they were trained. We trained them to come to the feeder, but they only encountered the obstacle once.
That's what I meant. Okay.
Yeah, and once they actually take part in the experiment, they're collected and put in a box. So they don't really, they're not counted again.
Yeah. Okay.
Yeah. All right. Okay, so this is a behavior that, so as you can see, we can conclude that it actually, size does affect the way you can avoid obstacles. But this is a behavior that requires good spatial resolution and sensitivity. But there's a lot of other navigation behaviors where you really don't need good resolution or sensitivity, for example. One of them is the ability to use celestial compass cues for orientation. So ants, for example, have this specialized ommatidia in the dorsal rim area of their eye, so that can detect polarized light. So when the sun's light enters the atmosphere, it gets polarized and it forms a pattern of polarization around it. And so these ommatidia are actually sensitive to that particular pattern, and then they use that to pinpoint which direction they should walk towards to find their nest, for example. And the thing about this is the number of these specialized ommatidia actually varies with the number of total number of ommatidia that you have. But there are ants that have as few as 20 ommatidia, and there are ants that have around 3,000 or even more ommatidia. But the proportion of these might also change accordingly. So how does that actually affect the ant's ability to use celestial compass cues such as these? So to answer that, we caught ants that were returning back to their nest in a dark vial, and then we transported them close to a cricket field, which was around 1 kilometer away from their nest. So they have actually never traveled there, and the cricket field is also surrounded by a water body. So we can be sure that they've never actually been there, and they don't have any familiar visual cues for them to navigate. And so in the middle of the field, we release them into this particular setup. And once they get out of that hole, they should be able to walk towards the direction of towards the direction where they think their nest should be, because they were actually walking back to their nest when we caught them. So this is what it looks like. They just come out, and most of the time they just dash off, and sometimes they stop there near the hole and then look around and then dash off, because they don't have any other familiar cues. They have to use celestial compass cues. So we repeated this across 8 different species that have 613 ommatidia to 21 ommatidia. And we plotted the paths taken by these all of these ants. And similarly, we also plotted the final heading directions of these ants as they exit the platform that you can see here. And so here the fictive nest direction for all of them is pointed upwards. And because like Sanjay just mentioned, to control for the stride length, we also scaled the distance at which we measure our heading directions as well for all the ants with respect to their head width. And so what we find is that almost all of them actually head directly towards where the nest should be. This is not something that we expected, because we thought they would have, they only have, one of them has 20 ommatidia and the other one has 613 ommatidia, and their specialized ommatidia must be obviously much less than what this particular, the largest ant has. But almost all of them, even after controlling for the stride length, almost all of them move towards the direction of their nest. But what you can actually see is that as the size increases, the paths also get quite wiggly. They're not quite straight as the larger ants. So what we think might be happening is that even though size doesn't seem to affect the overall ability to orient using celestial cues, it might be affecting the way in the precision and the accuracy at which they can detect this particular cue. And that's what makes their paths really go windy. And they're also slower. I haven't shown the data, but the smaller ones are also slower as well, even after controlling for differences in stride lengths, all of them. Right, so to answer.
One question, sorry.
Yeah.
So do we know about their detection of polarized light?
What do you mean?
So when you say celestial cue, one of the things that they can do infer is the polarization pattern, right? So you have like a gradient across the sky.
Yeah.
Since you have a clear gradient which has a very particular pattern, regardless of how many sample points you have from that gradient, you can still say which direction is what, right?
Right.
So then that kind of explains this, right? Whether you have more ommatidia or less ommatidia. Since you can sample that gradient, you can orient yourself to that.
Right, so that is correct. You can orient, but you need to be able to detect the pattern as well. So if your sensor is so weak, then it might be hard for you to precisely know this is the direction that the polarized pattern is showing me. And so in addition to polarization, something that I didn't mention is so they might actually be using a lot of other celestial compass cues as well, which which includes sun's position as well. And other cues like chromatic differences, chromatic patterns in the sky. Yeah, I cannot specifically say they're using polarized light in this case.
Okay.
Yeah. Right. So even though we find that size doesn't actually affect the way they can, their ability to use celestial compass cues for orientation, it is possible that their precision and the accuracy at which they detect a particular cue might be affected, and that's what explains differences in path straightness and differences in speeds as well. Right, so this is all the ant part. I've also done work on comparing how brains of different sizes invest in different neuropils. So whether relative investment in one particular neuropil actually affects the relative investment in other neuropils. For example, relative investment of sensory cues, how does that correlate with the investment in higher order processing cues given brain size? So I'm not talking about that right now. If anyone is interested, we can talk about that later. But I'm going to change the gears here and talk about the work that I did last year. So again, keeping the same theme of miniaturizations, we wanted to understand how miniaturization affects fruit fly vision. So one of the lab members, Pablo, he was actually able to isolate a third stage larva of fruit fly and starve them. And that actually creates size variation, the huge size variation that you see in Drosophila. But actually in the wild, fruit flies actually have really wide range of body sizes, as you can see. So it's quantified body length and eye diameter. And in the wild, there is a huge variation in body length. But because we feed them a lot in the lab, they get unlimited food. All of them tend to be larger in general. So if we, in the lab, if we starve them, we again create slightly more variation and smaller individuals, especially in the lab. And so he was able to do this, and then he measured, he estimated rather, how acuity and sensitivity varies with eye area. So what you can see is both acuity and sensitivity does reduce with reducing eye size of these flies. But what you can also see is that the rate of reduction in sensitivity is much faster than that in acuity. And again, as I've pointed out before, it's always important to check beyond the optics of the eye in order to look at what the actual measures are. So for that, we used a behavioral technique where so we have this virtual reality arena. What happens there is, so what's happening here is that you have 4 mirrors and then a cube in the middle. So you can project stimuli onto these mirrors, which gets reflected onto the cube. And inside the cube is a back projection material. And then inside that is sits a tethered fly that is free to yaw. So you can just, it doesn't roll or pitch, but it's just free to yaw. And we measure the response of these flies to different moving gratings again. And so the way we measure them is that we use an infrared light setup, which is shown from the top. And then you measure the amplitude of left wing and right wings as they move their wings in response to the stimulus. And then you can, so as they move their wings, it casts a shadow on the photodiodes, which you can actually measure. And that's how we quantify their turning behavior. So if they're not turning, there's not much difference between left minus right. But if they're turning in either of the directions, one of them tends to have higher amplitude than the other. And so that's how we know that the fly is actually responding to the stimuli. And so, so I've just plotted the same thing here. So this is basically left wing amplitude minus right wing amplitude. I'm going to be using that particular value in all other plots that I show from here. Right, so what we did was we measured 3 different abilities. So one in addition to what I've done for ants. So we changed the contrast of the gratings, and then we measured the response of the flies, the wing beat amplitude differences of the flies for smaller and larger flies. So we measured the body lengths and then distributed them into 2 different groups at the median. So the smaller flies are shown in green here and the large ones are in black. And then we also measured the spatial frequencies, response to the spatial frequencies. And in addition, we also measured response to temporal frequencies as well. Right, so what we see here, let's go from here, from the resolution. What we see here, so plus mark means that the wing beat amplitude is not, is significantly different from 0. That means they were responding. So smaller eyes have lower spatial resolution than larger eyes. That's what this means here, which is expected, which is what we find in the optics measurement as well. But what is surprising is that if you look at the contrast sensitivity, we don't really find difference in the contrast sensitivity between larger and smaller eyes, even though we saw that contrast actually reduces much faster than acuity when we look at the optical measurements. So one way this might be happening is that they temporally summate. So which is basically like a long exposure photography. So you're newly summing visual information over time. And so that helps you increase the contrast of the image that you see. So that increases your contrast sensitivity ability. But one drawback is that because you're temporally summating, your temporal acuity would reduce. And that's exactly what we find with the smaller flies. They seem to sacrifice their temporal acuity ability to recover their contrast sensitivity that they lost at the optics level.
This is very interesting finding. Also relates to flight. You would not see temporal summation in insects that flew fast. Drosophila, on the other hand, hover a fair bit, which means that temporal summation is possible because they are so steady relative to their environment when they're flying.
Yeah.
And these are also insects that seem to do very well under, indoor conditions, for instance, where light levels are pretty low. And again, under those conditions, they would, they would be required to temporarily summate a little more than they would in the bright outside world.
Right. That actually goes to my next topic very clearly. So you're right. Yeah, like even though we find that they lose sensitivity, they actually regain it by temporal summation. But eye size is not the only thing that affects your signal to noise ratio when it comes to vision. So the other thing that you just mentioned is the light intensity. So as it gets darker and darker, your signal to noise ratio is also affected. And similarly, if you're a fast flying animal, then again, your signal to noise ratio is reduced as you fly faster. So we tested one of them. So we wanted to understand how smaller flies, so how the visual abilities of smaller flies change with light intensity. And so we repeated the same experiment, the same setup at different light intensities. So the orange one is smaller flies and blue is larger flies. And so if you concentrate on on just this one, so all the patterns that we find here kind of confirm what we found in the other study. But as it gets really dark, around less than a lux intensity, smaller flies actually tend to lose their spatial acuity altogether. So they're not really responding as much as the larger flies. And similarly, smaller flies also tend to have lower, so minuses, sorry. So in the previous plot, we used pluses, but in this plot, we use minuses. So minus just means that the response is not significantly different from 0. So the smaller flies didn't respond as much in the darker conditions. And similarly, they had reduced contrast sensitivity in the darker conditions compared to larger flies. And similarly, again, temporal acuity is also lower in smaller flies. So again, if you're a smaller fly, you already suffer from reduced visual abilities. But if you're in the dark, again, your abilities are further affected. So what exactly do you do in order to cope with these? One of the possibilities is that smaller flies may not actually fly or forage at that really dim light. So they may actually start foraging when it's really bright. And the other possibility is that when they're in their free flight, so all of our experiments are done in rigid tether flight arena. When they're in free flight, they might actually slow down so that they can collect more information and fly and do their whatever behavior they're doing. So the other thing might be that they may pay attention to certain features that are more prominent to them. So they, in the darkness, if you're a smaller animal, you might actually pay attention to certain features in the surrounding more than the others. So all of these are alternatives that we are currently looking at. So we are trying to compare how they fly in free flight mode at different light intensities and with different environmental cues as well. With that, so to conclude everything, to come back to the miniaturization question, it can be studied across taxa, and we need to study how miniaturization affects visual abilities in different insects. And most likely that we would find, just like what we found in ants and flies, we might find unexpected adaptations to being miniature in different flies. And especially when it comes to this kind of variation that we find in different orders of arthropods, it would really be nice to have more data in, say, lepidopterans and coleopterans and other dipterans as well, to see if they really have other unexpected adaptations to having a smaller eye, depending on the ecology that they live in or the behaviors that they have to perform. And to answer how miniaturization affects behavior, we shouldn't just be counting the number of behaviors. We shouldn't also be just looking at whether they show a particular behavior or not. Rather, we should be looking at the precision and accuracy at which they can perform a certain behavior. And if we find that some smaller species have similar abilities as bigger species, we can further look into how are they being so efficient with having such smaller sensory structures and still be able to perform the behavior as good as larger insects. And another thing that I think would be interesting to study, and I don't think has been studied, is to look at how size affects motor response in different insects. Does it really make them as efficient as larger animals? Something that would be really nice to answer as well. Right, so with that, I'm going to thank all of you for this opportunity, and thank all my supervisors and lab members. And I can take more questions.
Thank you, Ravi. That's a fabulous talk, very thought-provoking. I mean, there's some very clear hypotheses that are being spelled out here. And, you know, I think the connection to flight, of course, is something that is very interesting and something that could be tested, for instance, in looking at just how much flies move around under lower and lower light levels. I mean, do they tend to hover more when light levels go down? Do they tend to hover less when they don't? You know, this is just something that seems like a very clear experiment to do, given what you've told us. So is there something that you guys are trying to do?
I have collected some of the data already. So we're trying to do some free flight experiments. So in addition to this, I'm also getting into multiple species comparison. So I would like to actually answer this from the perspective of phylogeny as well. So that's why I really like the paper that came out where you looked at different species and then looking at the phylogenetic context as well. And I also want to be able to see what ecological variables actually affect the way they fly in free flight.
But do you feel like phylogeny really would play a big role in this, beyond sort of the basic, because some of the, I know, for the flies that you've looked at here, they're the same species.
Same species, yeah.
So, you wouldn't have anticipated differences that were size related if you were just going by phylogeny.
Yeah, no, I meant not in this particular species, but looking at species that live in the desert, like Mohavensis, Drosophila mohavensis, and a few other species of Drosophila. So they have completely different visual ecology. And so one of the good things about this is that you can actually make size variation in the lab. You just have to starve them, or you just have to increase the temperature, and you see a large variation in size. So I want to see how they actually cope with being small if they have a different ecology, visual ecology. Because in addition to eye size, they also have other selection pressures adding on to them. Yeah, so that's what I meant by phylogeny. And so, yeah, the point is not to see if they're phylogenetically different, it's just to control for the phylogeny. So in this case, I didn't have to control for the phylogeny because it's the same species.
Yeah.
But it would be nice to like compare, but control for phylogeny as well.
Yeah, Partha and Maitri have questions. Maitri first, Partha next.
Okay. Hi, I had a couple of questions. The first one was regarding what you mentioned about size affects motor command. The one you want to address. So what I wanted to ask is, in what sense are you talking about this? Are you talking about the input coming in, which changes because the size of the sensory structure has itself modified, or you expect a different difference in the connection made to the motor centers?
At this point, I'm not really thinking about the mechanistic way of differences, but what we can actually, to start with, do is if you have a certain behavior where they have to use motor response, I'm going to give an example of, say, a spider making its web. So it's a motor behavior, right? So they're using their legs to build the nest. And so if you have a really small smaller spider, how exactly does that affect the way you make your nest, the way you use your motor organs to build the nest? I don't really have a concrete example for insects right now, but it's just something that I wanted to mention because you guys mentioned a lot of you work on motor response, but yeah, so you might actually know better than I do.
Okay. The next question I had was regarding the other project you were talking about, the investment made in neuropils.
Yeah.
I was just curious about how would you test it or measure?
Right. So we have actually done, so I've done a meta-analysis for this one. I collected neuropil volumes that have been published for different insects. I think I was able to get around 70 or 60 different species of insects. And so if we have the total brain volume, and then if it also has brain volume of say antennal lobe and optic lobes and mushroom bodies and central complex, for example, if you pull them and put them as a higher order neuropils, and if you pull these two and put them as sensitive neuropils, so we can look at the relative investment in these two with changing brain size, for example, and then control for the phylogeny again. So that sort of gives you, that tells you if you have a fixed volume for the brain, how exactly are you investing in different neuropils?
How much would you allocate for different neuropils, and
right.
What would you choose over the other?
Yeah, and what would that depend on? Would that depend on the ecology, or would that depend on diurnal nocturnal state? Because if you're nocturnal, maybe you invest more in antennal lobe more than the optic lobe, or maybe it's the other way. So yeah, so that's how we're doing. But Another student in Ajay's lab was also looking at not exactly this particular question, but he also stains the brain of different ants that are nocturnal and diurnal, and then he uses Amira software to like quantify the volumes, and then once you have the volumes, you can basically answer a lot of questions.
Right. Post hoc test other hypothesis.
Okay. Right. And in addition to that, I've also looked at relative investment, how it depends on flying and walking mode of locomotion. And so I would expect, actually we find that optic lobe investment is higher in flying insects, obviously, because they're using vision. And then similarly, we looked at how, say, mushroom body volumes differ with flying and walking insects, and in nocturnal and diurnal insects as well.
I see. Thank you. It was a great talk.
Yeah, Ravi, it's a nice talk. So I have two questions. One is when you looked at the ants, and so all from are from a similar habitat where they walk, like it's not like some are arboreal, some are on the surface. So all of them.
Yeah, all of them are actually within like a really small area as well. Maybe so it was just beside the building where I worked. So it's all in the same habitat, basically.
Cool. Okay. So the second question is about your last portion of the talk. It was very interesting because it's happening within the species, and is also during development diet restrictions. So the size changes or varies. So so though they seem to retain their temporal tuning, right? So they so is that can you just comment on that? Is it kind of developmental, or is it something they have to, with days they get better? Is that the tuning would change or as they spend more time in the environment, or is it the developmental where the tuning is just retained?
You mean, what kind of tuning do you mean? Is that the slide that I have right now?
The temporal tuning part? So, which is...
So the temporal acuity differences, you mean? I don't think development has to do with this. Once they have the eye developed, so once they develop to become an adult and they have the eyes, and so what I'm trying to say here is that they may be temporally summating the images that they have. So it's just like long exposure photography. So you have an eye, you have your images, but then you have one image at one point of time, and then another image at another point of time. You put them together, and that's temporal summation, like very crudely.
I understand that, but that is the property they have not lost, despite that size variability, right? So,
I'm sorry, I didn't understand the question then.
So the size variability. So when you compare the small flies and versus the large ones, right? So that's what you did, and then what you found, if I'm correct, and then the temporal tuning was retained to a large extent. Is that correct?
Contrast sensitivity was retained.
Sorry, the contrast sensitivity was retained, but so I was wondering, is that something they have to? So something to do with developmental thing because it is compensating for one, or is it something they have to get exposed and as it spends more time and then this develops? Is that something you guys, I don't know.
I cannot really comment on that, but I doubt that it is to do with development, because these are, so we typically test them 3 or 4 days after they've developed. So these are all similar aged animals as well. And so if at all, if it's anything, it could just be the difference in their visual system. But I can't particularly comment on the development part. I don't know.
Okay.
Any other questions?
Dinesh has a question.
Yeah, so I really like the approach of, I mean, this trying to figure out exactly, you know, what is lost when the miniaturization happens and what is retained is quite nice. And I think it is a really hard problem to tackle, simply because there are so many dimensions involved, right? One, you've already pointed out the number of behaviors that can be performed, which can change when the size goes down, the precision, and and of course there's learning. How flexible is the nervous system to learn new behaviors or new aspects of behaviors. So I was wondering how, it's very clear to me why initially for your PhD you chose ants, simply because that gives you all these in some sense, right? It gives you at least these three dimensions which you can play with. I was trying to think where else could you apply this in a more restricted manner. And one place is honeybees, right? You have Apis mellifera, you have Apis florea, which are so different in their sizes, but they
Right.
still forage, they still remember, they still, at least, very loosely seem to follow similar rules, right?
Right.
So has this been looked in deeply? How do they compare? Like,
Right. Yeah, that's exactly what I want myself and others to do, is to actually study these across different insects. And I'm sure there would be a lot of unexpected adaptations that are not seen in other species. And like you said, if you can look at the similar kind of questions in bees as well, and find out different adaptations, so that's exactly what would be nice. And yeah, so I think that would be nice, but I don't think it has been specifically studied from the point of view of miniaturization.
Dinesh, I think there's some bumblebee work from Ravi where they have looked at individual variability in flight performances that they can attribute to the size.
Right.
So the size seems to be explained why the individual flight performances are different to an extent.
Are you talking about Sridhar Ravi's paper?
Yes, yes.
Yeah, we actually discussed that paper yesterday. Yeah, so it's an interesting question. It again involves obstacle navigation.
Yeah.
That's the paper that. Sorry.
Right.
Yeah, if that's the paper that you meant to mention. But yeah, so there are some studies again in bumblebees and Gavin Taylor and some people in Lund are looking at bumblebee size variation and differences in vision. So I forgot about that. Thanks for reminding.
Is Gavin still at Lund, or is he transitioning out or something like that?
I think so. From last time when I heard,
He's not currently in any position.
Yeah, not that I'm aware of. I think Emily was looking for some new postdocs, so I don't know if she found me. But she's another person who is looking at this kind of variation in bumblebees. And then beyond that, yeah, there isn't many people right now looking at these kind of things.
Interesting. So any thoughts on how you would pool this sort of information? So now there are so many dimensions involved, right? There's Partha was saying flight stability. That's one aspect of it. Then maybe how the search area changes, or how quickly, how many trips to the flower would be required to remember it or transfer the message. So how would have you thought of how, let's say, you do these? Each experiment is a very precise question, but the whole aspect by itself is multidimensional, and you have to somehow fit it in and come up with a So have you thought of this?
Right. Those are all good points. I have actually thought about different behaviors, and in addition to this, and learning and memory is one of them that I really wanted to answer as well. I did some more experiments, which I didn't really talk about here, is to look at how well they learn a landmark. So if you give them a landmark outside their nest, they tend to learn it and use that to pinpoint where their nest should be, because so most often some of these ants have a nest which is just a tiny hole in the ground, and they have no way to tell where it is. And so they tend to use landmarks around if it's a tree stump or something like that. And so I was trying to understand that. It was really tough to answer this question. I was working on bull ants, and I was supposed to work, so they're nocturnal. So they're nocturnal, they go out to forage, and then I have to test them in the morning, early in the morning. That's when they return back to their nest. Yeah, and I had to mark the same individuals and follow the same individuals. In that process, I lost a lot of ants because sometimes they get eaten while they're foraging. So I ended up having like 10 ants or something. So I didn't really like the data that I had. I didn't really like, as in, I couldn't answer the question that I wanted to answer with just that 10 data points. So yeah. But that's something that would be really interesting to study, looking at the learning and memory differences in different insects with size differences. And also when it comes to flight, there's so many things that one can answer. For example, like Sanjay mentioned, hovering behavior is one of them, whether they turn more or whether they saccade more, depending on the size of the eye. And yeah, how well they can actually land or how well they can avoid something mid-flight, and all those behaviors that can be studied. Yeah, I'm thinking of doing more in that direction.
That's great, Ravi. Really enjoyed this talk, and
thank you so much.
Yeah, this was good fun.
Yeah.
So good, and you'll continue to join us for lab meetings.
Sure.
Yeah, great. I mean, there's no like, you can be part of the discussions.
Yeah.
You know, data, things like that. I think it would be wonderful to have you join whatever. If you're really interested in excruciating details about animal breeding, then you can be part of that too.
I would actually love to know about the fairy fly, the mimarids that you have.
Yeah, well, we don't have fairy flies. We have yeah, trichogramma.
Yeah. Okay.
Yeah. So
yeah, I would love to know more about that, and also the motor response. I actually intentionally put it there because you guys work a lot on motor stuff, and if you really find size variation in the animals that you study, you can easily test some of the hypotheses.
Yeah. So Girish has been ominously quiet. But Girish is the one who's actually working on the trichogramma and yeah. If anyone, I think he can do electrophysiology on these.
That would be really cool. We don't really know anything about. We don't know a lot of things about this.
Yeah. No. He's sort of been able to tether the flies and so on, which is just bizarre. I mean, I never thought that was possible. So
how exactly? What do you tether it to? What kind of?
Girish.
So I use these glass capillaries or electrodes that these guys use for electrophysiology. So I just put a tiny drop of Fevicol at the end and just stick it to the thorax. I can show you some videos later.
Sure.
Yeah.
I would be interested. If you can email, that would be great. I don't know if I put my email anywhere.
Yeah.
But yeah, it would be cool. And I would love to know more about it. So we can chat about it later.
Sure.
All right.